hypotheses. We do think that also related to Bruce Ellis' work that, and actually there's a lot of research in animal studies, okay? So when I mentioned we don't know that in humans, okay? But evolutionary ecologists actually, telomeres is a very hot biomarker. And they have a lot of kind of life history kind of processes that they did document that telomeres can function as this kind of a factor that can explain that. So there is kind of indication that it might even regulate earlier preorbital maturation. So it's a good question. We don't know, but we hope to be able to test that if NIH will give us the money. We'll see.
I hope they do. Well, great. Let's stop there and take 5-10 minutes for a coffee break, bathroom break, whatever you all need. Let's meet back here in about 5 or so minutes.
Go a shorter break because we need to stay on schedule because we have speakers that need to be on planes this afternoon. So sorry for the short breaks, but please grab your coffee and come back. Thank you so much. Get some glasses.
You can just use this because there's multiple screens, and so you can only get one.
I'm going to go grab a glass.
Take your time. So far, so good. Don't go away. So far? That was good. I think I'm just going to plug in. Oh, there you go. Of course, this thing is all twisted. There we go. Outstanding. We are all good to go.
It's great. It's great to be a part of it. It's great to have everybody come and talk about their work. I'm doing work with genetics and epigenetics, so not so much telomeres, but more methylation. Sequencing it. Right. And so I think it's a credit to the speakers in that they're able to talk about these complex biological processes in a way that is understandable, I think, to everybody. So.
Well, we're going to put an end to that. Yeah, absolutely. Absolutely. I'm aiming for incoherence, and I'm good at that. Chad, do you have an affection for long introductions?
Well, I do. I sort of
Okay.
like doing that, but
Okay, no.
I don't have to. In fact, I should probably not, given that I was sort of, negligent on my duties to keep people on task.
There you go. Michael Meaney, here we go. I'm all for it. There, we saved a few minutes.
Okay, we're going to go ahead and get started with our integrative speaker for this session. So as a way to help keep me on task in terms of doing a better job with time management, me and the presenter have decided that we are going to forgo with the longer introductions and just essentially say, here's Dr. Michael Meaney.
There we go. Or gets none anyway. So. Look, you know what? You're either good or you're not, right? So forget it. So I'm trying to do something a bit different. I was trying to put myself last night in the chairs that most of you have been occupying. And first thing is, I wanted to just as a global comment, yesterday's presentations were, absolutely top drawer. It's an amazing thing to come to a meeting on childhood adversity and see the sophistication conceptually as well as the data that you got yesterday is really quite impressive. It was a real joy and great credit to the organizers as well as, of course, the speakers. So it's my goal to put a complete end to that. So what I thought, you got so much good science yesterday, I thought I was going to try to do something a little bit different and say, okay, now how do we close gaps here with respect to creating this much science and then putting it into a place where it can be delivered with impact? So I thought I'd do is start you off with a model, something where this has been incredibly successful. It's an example of where biomedical research has come to the rescue of a highly vulnerable population, old men, which I think much more fondly of these days. And one of the reasons why you see so many of these characters around is essentially because between 1960 and 2005, biomedical research produced a 75% decrease in the risk of premature death due to heart disease. 75%. So just stop for a second and imagine that in the year 2035, somebody's up here, maybe me, saying, no, okay, not me, but saying we've produced a 75% decrease in the risk from mental health. Like, just ponder that. So, okay, so the American Heart Association did it. So how did they do it? Well, they started off by, first of all, recognizing risk factors. The Framingham study, which many of you, of course, will know about, small town outside of Boston, turned out to be the heart of this tremendous advance in medical research because it allowed people to say, well, okay, what predicts, in a classic epidemiological sense, what predicts an increased risk for heart disease? It allowed people then to follow up from that and to look at the links, biological, psychological. We don't care right? We want predictors. So of the nature of that measure is we're agnostic with respect to what it turns out to be. But work on understanding the links that form the basis of the relationship between the risk factor and the outcome is critical, because that is really how you then take a particular measure and scale it down to the individual. And of course, that's where the American Heart Association and those who follow its guidelines have developed algorithms. So you go to see your internist, he punches in a list of factors and tells you, here's your risk of heart disease, particularly stroke. So prevention then focuses on individuals. And our biggest challenge is to go from targeting conditions, poverty, childhood maltreatment, birth outcomes, to targeting individuals. That's where we need to get to. And so that's sort of what I wanted to walk through a little bit about how there are different ways that we might think about that. What you've had over the course of yesterday and of course in this morning's early sessions is clear evidence that childhood adversity is biologically embedded, that it creates an impact at the level of brain structure and cellular function that can potentially form the basis of a link. What you've also been hearing about is that the impact of adversity is genetically moderated. You heard that from Christine yesterday. You heard that in Dr. Bradley's talk, and many others. And so we know that conditions, targeting conditions alone, is going to be insufficient, because in the classic CASPI study, which has been replicated at least a number of times in both form and in function, is that there are multiple individuals who, at least with respect to depression as an outcome, seem to be largely resistant, and others, of course, that are considerably vulnerable. And Master and I were talking last night, like, while we tend to focus greatly on these pathways, we tend to ignore what's going on here. I mean, you have a percentage. By the way, LLs are more plentiful than our SSs, right? So in terms of just simply numerical, most people here are not showing what we would assume is the predicted outcome. So what's going on? What is the biological basis of this particular outcome? So I'm going to walk through just a little bit about what is our major source of affection. And you heard that from Kieran this morning, and that is epigenetics. I want to give you one reason, although I'm going to drag it out, as to why I think epigenetic marks may be useful as predictors. And largely what I'm going to talk to you about is the idea that they could be convergent mechanisms. They could be a signal that is integrative at both the level of relevant environmental exposure and of genotype. So our work in the field of epigenetics was inspired, as I'm sure just about everybody's was here, in terms of epidemiology, and in particularly looking at the profound effect of early family life on multiple health outcomes. We have, again, tried more recently to start to look at the divergence here, how it is that individuals exposed to what seem to be comparable levels of adversity show the absence of these particular outcomes. We started by simply trying to define the biology of this, and thank you, Bruce. You did a charming job of summarizing our work yesterday. What we tried to do was to really ask the question about how it is that social adversity becomes biologically embedded. We used a rat model in which we look at variations in maternal care. It may surprise you to know that rat mothers do differ in the way they treat their offspring, and it's quite stable. They vary in the behavior that you see pictorially represented here, and this is this licking and grooming behavior. The frequency with which mothers lick their pups in the first week of life is associated with many phenotypic outcomes that endure into adulthood, in some cases late adulthood. They can be at least in part, if not completely, reversed by cross-fostering, which tends to suggest that it is the care of the mother that is driving this phenotypic variation. And we built on this particular model to develop what is known as the environmental epigenetics hypothesis. It is simply to state that profound, biologically relevant social events such as maternal care alter cellular activity throughout the body, leading to potentially epigenetic changes in epigenetic marks that regulate gene expression and thus broader measures of phenotype. We looked at that, and you heard this from Christine yesterday. We've looked at that largely in form of the hypothalamic pituitary adrenal response to stress. And essentially what emerges from these studies is that when we look at the offspring of high-licking mothers, they produce more glucocorticoid receptors in the hippocampus. They have more efficient feedback regulation over CRF from the hypothalamus and ACTH from the pituitary. And their glucocorticoid responses are more modest by comparison to animals reared by low-licking mothers. And we've used both pharmacological and genetic manipulations to show that it's the difference in the glucocorticoid receptor that drives these differences in pituitary adrenal responses to stress. We've then wandered into the field of epigenetics, and what we're intentionally trying to do is to simply ask a question that's been lurking in the literature since the 1940s and the 1950s. How is it that early experience produces a sustained change in cellular activity? And our target here, of course, was the glucocorticoid receptor gene. Now, this was interesting to us because we thought at the time epigenetics was swimming a little bit upstream to what was the massive enthusiasm for genetic variation that was, of course, launched with the Human Genome Project. Needless to say that when the Human Genome Project was launched, we anticipated that the many hundreds of thousands of different genes would explain the complexity of our wonderful species, right? And that we could then appeal to variation within those many hundreds of thousands of genes to understand phenotypic variation. Well, then it turned out, okay, so maybe it's not a few 100,000. It's got to be 100,000. I mean, the damn carrot's got 19. Well, it's not 100,000, it's about 27. And these guys over here, this guy, by the way, is killing himself laughing. I don't know if you can see that. That's a water flea. He's just found out he's got 31,000 genes, and you have 27,000. You think that's bad? Your genome has about 3 billion base pairs. This lungfish has like 40, 50 times that many. So, I mean, we're rather pathetic at the level of DNA. So, essentially, we better hope that the following is quite true. And no longer is this statement only true for males. This, we hope, is the answer to what you do with it. It's about epigenetics, and as Kieran described, essentially what occurs is that epigenetics it's the biochemistry of gene transcription. That's all it is, really. It's a series of biochemical modifications, some of which occur to the DNA, some of which occur to histone proteins, which collectively are referred to as chromatin. And they either open this up, so transcription factors can bind and turn on genes, or, as Kieran said, they close it, in which case transcription factors can't bind so easily and genes tend to be silenced. And that's essentially epigenetics. So what we're interested in is variation, because the beautiful thing about epigenetics is that, well, look, this was the Human Genome. It was great, by the way. You'll learn why I love the Human Genome Project later on, but it was a bit of a sales job, right? Because, I mean, after all, somebody turned around to, I'm afraid, your Congress and said, we want lots and lots of money because we're going to understand how polymorphisms can explain diseases, right? And nobody thought up and said, hold it, muscle cells, brain cells, they're all like really different, but they have the same DNA. Isn't that kind of a problem? Didn't seem like it was. Anyway, what turns out, of course, is the way in which you produce phenotypic variation is you do work from a common genetic template, but you produce multiple epigenomes. These are cell-specific. And what, of course, is there's the potential that then a common genome can, in fact, be translated very differently in different cells, in part due to environmental exposures. So we asked that question, and indeed, many years ago showed that maternal licking does indeed alter intracellular signaling pathways in areas like the hippocampus. And this remodels epigenetic signatures, including those at the glucocorticoid receptor, and in particular, differences in methylation. So this was just kind of a proof of principle. It suggested to us that the social event can indeed bring about these changes in the epigenome, and that they can be meaningfully associated to functional outcomes. In collaboration with Gustavo Turecki and others at McGill, we went on to show comparable effects In humans, we use post-mortem human hippocampal tissue, from which, thanks to Gustavo's wonderful forensic phenotyping, we had information about mental health as well as developmental history. We looked again at the same region of the glucocorticoid receptor and found in this particular case that, and I'll spare you all the slides, that a previous history of childhood maltreatment associated with an increase in methylation. And so there's increased methylation of the glucocorticoid receptor gene amongst individuals who have a history of abuse. And Christine and others have shown, Michael DeBellis, et cetera, that abuse associates with differences in pituitary adrenal activity. Now, okay, so then we get to something that takes us into the potential world of translation. Since we published that paper, which was 5 years ago, there have been a series of papers which have looked at childhood adversity in multiple forms. They've looked at the same genomic region, that is the promoter of the glucocorticoid receptor that we defined in rat and later human hippocampus. But what they've done is they've looked at peripheral cells. Those are cells that one can rather easily obtain by comparison to brain cells. And so they've looked at leukocytes or at buccal swabs, such as what Kieran described earlier. In other words, they look at accessible material from living subjects. And what they found is essentially the same relationship. So Gustavo and I actually performed a meta-analysis, which is in press now. It's actually out, I think, in Biological Psychiatry. We found 14 studies which had looked at early